Hello, good evening, everyone. Welcome to today's webinar. And today's webinar is it is about the states of matter and also the properties of the drug molecules. So here, before actually starting this particular webinar, we are going to discuss about what actually the states of matter is and how the various properties of the drug molecules are affecting at each and every stage of the drug manufacturing or even the drug when it is showing its effect, right? So coming to the introduction of this particular topic. So here, first we are going to see what is the importance of states of matter in the pharmaceutical sciences. So there are three important primary states of matter here. Like we have solids, liquids, as well as gases. So these are the three primary. We have another states of matter also, but however, we do not have its importance at present in the pharmaceutical sciences. So they play a very critical role in the design as well as delivery of the pharmaceutical products. Like for example, if you take the solids, they form the basis of most dosage forms like tablets, capsules, powders, which provide the stability as well as the convenience. And then coming to the liquid, you have various dosage forms like syrups, injectables, emulsions for faster absorption and ease of administration. And also we have various gaseous substances which are found in mostly inhalers as well as the aerosols, which are used for targeted drug delivery, especially to the respiratory system. So understanding these states of matter, it always helps in predicting the drug behavior, manufacturing process, as well as the product performance. So these state transitions, they are mostly helping predicting the drug essential in the drug formulation process, like freeze drying, spray drying, as well as in order to enhance the stability as well as the bioavailability. And also this has a particular relationship between the physicochemical properties and the drug performance. So what is the relation between the physicochemical properties and the drug performance is? The physicochemical properties that we have is the solubility, polymorphism, vapor pressure, and latent heat. So these are the various physicochemical parameters that we have, and we discuss about each in the following slides. However, the solubility, it affects how well a drug dissolves in the body fluids, which influences the absorption and finally the bioavailability. And then polymorphism, it has different crystalline forms. Vapor pressure, the drug tendency to evaporate. Again, we'll be discussing about the critical point and the phase diagrams here. And then latent heat, it is the energy requirements during the phase transitions. Okay. And then also we are going to see about the applications in the drug formulation as well as the stability. So by having the knowledge of these states and states of matter, it always helps in selecting the appropriate dosage forms. Like if you want a stable and long lasting formulations, you have to go for the solids like tablets. If you want for ease of administration and quick onset of action, then you have to go for liquids. Then if you want to have a formulation that is ideal for conditions that are required, either rapid absorption or localized drug delivery, for example, like asthma, you have to go for gases. So like this, we will be having various applications of these particular states of matter in most of the drug performance that we are seeing today, right? So moving to the next slide, as I said, we have four states of matter. The first one is solid, gas, liquid, and then plasma. And however, we are not going to discuss about the plasma right now because it does not have any crucial or it does not have any role in particular pharmaceutical sciences. So what are the solids? So you must have studied in your 6th or 7th standard itself what are solids. Why, madam, why we have to study it again? Because here the solids, liquids and gases in relation to the pharmaceutical products you are studying, right? So here they are characterized by the fixed shape and volume because of the tightly packed molecules in a structured arrangement. And we have two types of the solids here, crystalline as well as the amorphous forms. So crystalline forms are nothing but the molecules which are arranged in a well-defined and orderly pattern, which we call it as a lattice arrangement. okay, which we call it as a lattice arrangement. And also they possess different melting points, and also each form they have a different stability. And examples here, the crystalline forms, you can take the general examples like sodium chloride, paracetamol, they comes under the crystalline solids. And then coming to the amorphous solids, they lack a regular arrangement, which results in the higher energy and solubility when compared to that of the crystalline forms, and they do not have any sharp melting point. So example here, you can take them as a itraconazole, okay, itraconazole, ibuprofen, they all belong to the amorphous kind of the products. So what is like these, when you know whether your drug product is of crystalline or amorphous, you can know about their stability, because the crystalline forms, they have a high stability and easy for handling, whereas amorphous forms, they improve the dissolution and the bioavailability. And then coming to the second one, that is the liquid type of the dosage form. So these have a fixed volume, but they take the shape of the container. They adapt to the shape of the container, because due to less rigid molecular interactions when compared to that of the solids. They flow like they have the flow nature. Especially we'll be using them in injectable syrups and emulsions. And also here, the viscosity plays a major role, especially in the liquids. The viscosity plays a major role in the pharmaceutical formulations. Examples include oral syrup solutions, injectables, emulsions, suspensions, where we want to deliver the poorly soluble drugs, right? And then coming to the gaseous, the state of gas, they have no fixed shape and volume. They are very highly compressible, right? They occupy the entire volume of the container. Pressure and volume are inversely related. Which law supports this? You would have studied already in your PP1. So it is inversely proportional. It is Boyle's law. Okay, so it supports the Boyle's law, which is important in designing the aerosol system. So we have the examples like medical gases like oxygen, nitrous oxide, aerosols we have, which we use in the asthma or COPD cases, right? And then plasma, as I already said, we are not discussing this as it does not have any relevance with the pharmaceutical sciences. Moving on to the next slide, we have the state transitions. That means, for example, even our state also transits, right? The state transition will be there for us also. Now we'll be very happy. Then our state will transform into very, very happy state, or even sad state, or even very dull state, like that, isn't it? So these transitions have certain names. Like when solid is being transformed to liquid, we call it as a melting. For example, melting of waxes, melting of excipients during the tablet or capsule manufacturing. So this melting, it always helps in understanding what is the thermal stability and formulation of the drugs like suppositories. You would have already prepared suppositories in your first year. Okay. And then the second transition is liquid to gas, which is called as a vaporization. So vaporization of the solids during spray drying or freeze drying, right? And also distillation process. And then it is always helpful. This particular vaporization, it plays a key role in preparing the inhalation aerosols and the solvent evaporation methods. And then solid to gas, it is called as a sublimation. Okay. So direct transition of a solid into gas, it is called as a sublimation. There is no mediator here. Solid to liquid, liquid to gas, we don't have. Directly from solid to gas, we have. It is called as a sublimation. So, for example, you must have seen camphor at your home, which we generally use in the pujas, right? So camphor in certain tropical countries, what happens? It becomes liquid because of the temperature that they have. It becomes directly into the gas. Okay, it evaporates. It reduces in its size because it has been evaporated. And also sublimation we'll be seeing in freeze drying, which utilizes the sublimation in order to improve the stability of the biopharmaceuticals. And then coming to the next type of transition that we have is the gas to liquid, which is called as a condensation. We generally call it as a cooling of gas. Okay, we generally call it as a cooling of gas is a condensation. So this is mainly useful in the recovery of volatile solvents in closed systems during the drug manufacturing. And also it prevents the wastage of the solvents, because once it has been evaporated, it has been collected. Again, that it is converting into the liquid, you're not wasting your solvent. You're not just leaving it to evaporate. And then the next one we have is the liquid to solid, which is called as a freezing. Everyone are very much habituated of eating ice creams, ice cubes. It is nothing but the freezing. So transformation of a liquid into a solid at its lower heat, as it loses the heat, it is called as a freezing. So this example in the pharmaceuticals, we can take it as freeze drying, which begins with the freezing of a liquid drug solution. And also it maintains the integrity of the heat sensitivity drugs. So this thermodynamic implications, we should also see about this thermodynamic implications in the formulations. That is, what is the energy changes during the transition. So here we have to talk about latent heat as well as the enthalpy changes. We have to talk about these two things. So latent heat is nothing but the heat absorbed or released during a phase change without changing the temperature. So we have two types of latent heat and all. We will discuss in detail in the coming slides. And coming to the enthalpy changes, it is nothing but the energy that is required to overcome the intermolecular forces during this state transitions. It is called as a enthalpy change. For example, endothermic process, we have melting and vaporization. Endothermic is heat absorbing. So it has to absorb the heat, and then only it undergoes some melting and vaporization. And exothermic process we have, which liberates the heat, where we can have the examples of freezing as well as the condensation. And also these state transitions, they have the role in drug stability also. Okay, they have the role in drug stability as well as the formulation. We have certain thermal analysis like differential scanning calorimetry, which is required to study the melting points and the phase transitions. And also it ensures the drug excipient compatibility, and also it predicts the stability, right? So this is what the state transitions are. And now we will be moving into the next topic called as a vapor pressure. So why we have to discuss about the vapor pressure here is, it has like we need to know about this vapor pressure in order to understand the evaporation behavior of the solvents, because every time we will be using the solvents in the manufacturing of the drugs, right? So what is the evaporation behavior of those solvents, we need to know. So what is vapor pressure? It is the pressure exerted by the vapor in equilibrium with its liquid or solid state at a given temperature. It is called as a vapor pressure. It always reflects the tendency of the molecules to escape from a liquid or a solid surface into the vapor phase. And higher vapor pressure indicates greater tendency for a substance to evaporate, right? And also it is always important to understand what is the evaporation behavior of the solvents and also the stability of the liquid formulations. And it is measured by using the manometric methods and the dynamic methods. So manometric method is nothing but which we determine the vapor pressure by using a equipment called as a manometer. Then it is called as a manometric method. So it is mostly used for non-volatile as well as a semi-volatile compounds. Like for example, measuring the vapor pressure of drug substances during the stability studies. Okay. And dynamic methods here, it always relies on the flow of gas over the liquid or solid to measure what is the rate of evaporation and the corresponding pressure, right? And then what is its relevance? What is the relevance of this vapor pressure is, the first one is it is going to predict the evaporation. And within the evaporation, it is nothing but mainly concerned with volatile solvents and then drug stability. Okay, so high vapor pressure solvents, they evaporate quickly, so that these type of the solvent will be helpful in spray drying as well as the solvent casting methods. And drug stability is like low vapor pressure compounds are less prone to the evaporation, so that they ensure the longer shelf life. So for example, the stability of essential oils in topical formulations, right? And then its relevance with the stability of liquid drugs. So it always, the vapor pressure always helps to identify the drugs and excipients which are prone to evaporation under the storage conditions, so that it prevents the loss of active ingredients or even the solvents, so that they preserve, or they they do not disturb the drug efficacy. And then we have the solvent selection. So with the help of this vapor pressure, the solvents with appropriate vapor pressure can be chosen for various processes, like high vapor pressure for fast evaporations, systems like film coatings of the tablets, and low vapor pressure for sustained release formulations, right? This is about the vapor pressure. And then followed by the critical point. So this critical point here, it is nothing but the temperature and pressure where the liquid and gas phases become indistinguishable. That means they become identical, forming a single phase called as a supercritical fluid. We call it as a SCF, supercritical fluid. So at this point, the substance it produces, or it exhibits very unique properties, like the density similar to the liquids, whereas viscosity and diffusivity, which is similar to that of the gases. And beyond this critical point, the substance cannot be distinguished either as a liquid or a gas. So that is a separate state. And this particular critical point is very useful to understand the supercritical fluid behavior during the drug manufacturing process. So it is important for the supercritical fluid technology, because the supercritical fluid is a substance at a temperature and pressure above its critical point, allowing it to dissolve the materials like a liquid and diffuse like a gas. And also it is useful in the drug extraction process as well as the particle size reduction. So how it is useful in the drug extraction? The supercritical fluids, especially like carbon dioxide, they are used to extract the bioactive compounds from the natural sources, like caffeine removal from the coffee, active ingredients extraction from the plants for the herbal formulations. In all this, we use the supercritical fluid technology. And also we use in the particle size reduction. This technology, it is mainly used to produce micronized or nano-sized particles, especially for the poorly soluble drugs. So for these types of the poorly soluble drugs, we will be using supercritical fluid technology in order to make them into the micrometers or the nanometers, because the supercritical fluid, it is going to dissolve the drug under high pressure, and rapid decompression, it causes the precipitation of the fine particles, right? So this is how the critical point is applied in both supercritical fluid technology as well as drug extraction and the particle size reduction. And then moving forward, the another property that we have is a polymorphism. So the polymorphism, it is the ability of a substance to exist in multiple crystalline forms. That means polymorphism will be exhibited only by the crystalline forms, but not the amorphous forms. So simply saying, the ability of a chemical substance to exist in multiple crystalline forms, where each type of the crystalline form have a distinct molecular arrangement as well as packing. It will not be similar to each other. So these crystalline forms or these polymorphic forms, they differ in the physical properties like melting point. They also differ in their solubility and also stability. So each type of the polymorph, they differ in melting point, solubility, as well as the stability. Right? And we have two types of the polymorphs, that is monotropic, where only one stable form will be there, and enantiotropic, where it is interconvertible form. That means the monotropic polymorphism, here only one crystalline form is stable under all conditions, whereas the other forms, they eventually convert to this stable form. Like a drug with a single stable polymorphic form, it is used for the long-term stable formulations. And enantiotropic polymorphism, the different crystalline forms, these are stable under different conditions. Like for example, the temperature or pressure. At one temperature, if the polymorph A is stable, then at another pressure, polymorph B would be stable. Okay. So a drug that switches between the forms, it depends upon the storage condition. So what is the impact of this polymorphism? It has an impact on the solubility, stability, as well as the bioavailability. How it is showing its impact on the solubility is like a more soluble polymorph, it increases the bioavailability, while a less soluble one, it always delays the onset of action. Okay, like the different polymorphs, as they have the different melting points, different solubility properties, it is going to affect the solubility. And then how it is affecting the stability, the stable polymorphs, they generally ensure the longer shelf life, whereas unstable forms, they degrade very faster. Example like instability in a polymorphic form, which leads to the loss of the potency. And another most important one is this polymorphic form, it is going to even change the bioavailability of the drug. Like, as I already said, and solubility and bioavailability goes always hand in hand. So polymorphs with higher solubility, they enhances the drug absorption and eventually the therapeutic efficacy. And the reverse happens if the solubility is less. So here we have an example called as the ritonavir polymorphic forms. So ritonavir, you know that it is an antiretroviral drug which is produced initially as a stable crystalline form. And a more stable but less soluble polymorph has been appeared during the storage, reducing its solubility as well as the bioavailability. So what impact it has shown? It has required reformulation to ensure that consistent drug performance is seen. So this particular example, it always highlights the importance of controlling the polymorphic forms during the drug manufacturing process. This is how the polymorphism is very much important during the drug manufacturing process. And then the next one we have is the latent heat. So the heat required for phase change without the temperature change is called as a latent heat, right? So it occurs because the energy is used to overcome the molecular forces rather than increasing the kinetic energy. And we have two types of latent heat here: latent heat of fusion and latent heat of vaporization. So if solid is being converted to liquid, it has a latent heat of fusion. And if liquid is being converted to gas, it has a latent heat of vaporization. So the latent heat of fusion example, you can take it as melting of excipients. Like you would have used the cocoa butter in preparation of suppositories, right? So that is called as a latent heat of fusion. And then latent heat of vaporization, evaporation of the solvents during the spray drying process. Okay, those are the examples for latent heat of fusion as well as the vaporization. And it is applied. This particular latent heat, it has major application in the freeze drying as well as the spray drying techniques. So freeze drying technique is nothing but here the drug solution will be freezed and then sublimed with the ice and directly into the vapor using the latent heat. And spray drying, here the liquid feed will be atomized into fine droplets and rapidly they are dried using the hot air so that leaving the solid particles. So these are the four things that we have seen so far here. And then now we are switching into the next topic called as a physicochemical properties of the drug molecules. So this is the second topic of our webinar here. So physicochemical properties of the drug molecule. In this, the first one that we are seeing is about the refractive index. So the refractive index is nothing but it is a measure of light bending when passing through a medium. Okay, so it is a characteristic feature of a substance and also it varies always with wavelength as well as the temperature. It always varies with wavelength as well as the temperature, and it is a characteristic property of a substance. That means it differs from one substance to the another substance, and it is measured by using a Abbey refractometer. Okay, and because this Abbey refractometer, it is the most commonly used device in pharmaceutical applications. Earlier, when we were doing B-Pharmacy, we had this experiment, but nowadays, I don't think so that you have an experiment of determining the refractive index using refractometer, right? So here what we do is a small amount of the liquid sample will be placed in the refractometer's prism, and the light will be pass through the sample, and the angle of refraction is measured. And the refractive index is calculated based on the angle and also known properties of the instrument. Okay. So what is its relevance with that of the pharmaceuticals is it is going to assess the purity. So with the help of the refractive index, you can assess the purity of the compound. And also if any deviation from the expected value, it may you may think that there are some contaminations or impurities that are present in the product. And also it is used in the identification of the solvents, because each solvent will have unique refractive index, which allows its identification as well as verification. So you can distinguish between the similar looking solvents like isopropanol, ethanol, right? So based upon the refractive index, you can differentiate which is isopropanol and which is ethanol. And also in general quality control. Okay, in general quality control also, this refractive index is used to ensure the consistency as well as the compliance with that of the pharmacopoeial standards. And then the second one that we are going to see about is the dielectric constant. So what is this dielectric constant? The ability of a substance to store electrical energy in an electric field, it is called as a dielectric constant. So it always indicates how well a material can reduce the electric field strength within its, when compared to that of the vacuum, right? And it is always used to characterize the solvent and predict their interactions with that of the drug molecules. And its importance is seen in solvent polarity determination. And here, two categories will be there: high dielectric constant, and we will be also having low dielectric constant compounds. So the high dielectric constant, it always indicates the polar solvents which are capable of dissolving both ionic as well as the polar compounds. The most common and favorite example here will be water. And the low dielectric constant, it indicates the non-polar solvents which is suitable for dissolving the lipophilic as well as the hydrophobic compounds. And the most common example we have here is the hexane, right? And also it is important in drug solvent interactions. So because here the dielectric constant, it influences the solubility, ionization, as well as the stability of the drug molecules. So when it is affecting the three major properties of the drug molecule, so it is always important in knowing what is its drug solvent interaction. Like for example, weak acids and bases, they ionize better in polar solvents like water, which enhances their solubility. So the examples we have for high dielectric constant is a water, and low dielectric constant is a hexane, right? And then the next property, physicochemical property that we have is the dissociation constant. So what is this dissociation constant? Like this dissociation constant is nothing but the measure of drug ionization in the aqueous solution, and it is generally determined by the equation called as a Henderson-Hasselbalch equation. So this Henderson-Hasselbalch equation is different for weak acids as well as a weak base. For weak acids, the equation is pH is equal to pKa plus log concentration of conjugate base divided by concentration of weak acid. And the reverse happens here. POH is equal to, that is for weak bases, pKb plus log concentration of conjugate acid divided by concentration of weak base. This is the Henderson-Hasselbalch equation for both weak acids as well as the weak bases. So how you can interpret this Henderson-Hasselbalch equation? For example, if pH is equal to pKa, then 50% of the drug exists as a ionized and 50% exists as a unionized compound. If pH is less than pKa, then the drug will be more in the unionized form so that it will have a better permeability. And if pH is greater than pKa, then it will be more ionized form, which is used for the better solubility. And its applications include, it always used in predicting the solubility at different pH levels, and also it is used in the drug absorption as well as the formulation. Okay. So how it is going to predict the solubility at different pH levels? The ionized form of a drug is more water soluble but less permeable across the biological membrane. So in that way, like for example, weak acids, they are more soluble in the basic environments, and weak bases, they are more soluble in the acidic environments. And also it is useful in drug absorption and formulation, like the unionized forms, they are more lipophilic and they cross the membrane easily, enhancing the absorption, whereas adjusting the pH of the solution or formulation, it favors the unionized form. It improves the drug permeability in the gastrointestinal tract. And also it helps in optimizing the drug delivery system also, because when you have the knowledge of this pKa, it helps in selection of buffers for the injections or parenteral solutions in order to have a stable and the drug with the efficacy, right? And then we are going to see about the properties that are related to the drug stability. Under this, we are going to see about two things. The first one is the phase diagram and the second one is the hygroscopicity. So the phase diagram is nothing but it is a graphical representation of a relationship between temperature, pressure, and the physical state of the molecule or physical state of the substance, right? So here it indicates the phase, the transitions such as melting, sublimation, as well as the vaporization. And it is always used in predicting the polymorphic stability. Okay, it is used in identifying the, predicting the polymorphic stability. Like the phase diagram, it always helps in identifying which is the stable polymorphic form of a drug under specified conditions. And also it is mostly used in the formulation design, like it helps in the selection of like processing conditions in order to avoid the undesirable phase changes. And also it helps in freeze drying optimization, like it is used to determine the sublimation point and also to design the lyophilization cycles for the thermolabile drugs. And the second one that we have here is the hygroscopicity. So what is hygroscopicity? The tendency of a substance to absorb moisture. Okay, the tendency of a substance to absorb moisture, it is called as a hygroscopicity. And we have two types of the compounds, non-hygroscopic and hygroscopic. The name suggests if it is not absorbing water, it is non-hygroscopic. And if it is absorbing the moisture and it forms as a hydrate, then they are hygroscopic in nature. So they are generally shows the impact on stability, because moisture absorption, it may lead to the chemical degradation like hydrolysis, as well as the physical changes, like it may lead to the formation of the lumps or as well as the crystallization process. For example, aspirin, it degrades to acetic acid and salicylic acid in moist environments, right? And then it also affects the storage as well as the packing conditions, because it requires a moisture-proof containers for the hygroscopic drugs, like desiccants should be used to protect the sensitive drugs like effervescent granules. So this is how these two are the properties that are related to the drug stability, the phase diagram as well as the hygroscopicity. And then the examples of state transitions in the pharmaceutical. So we have seen some transitions in the beginning of the webinar. So what are the examples, pharmaceutical examples that we have for these phase transitions? The first one is the solid to liquid. The most important example that we have is melting of excipients during the tablet compression. So during tablet compression, certain excipients of the formulation, they may partially melt due to the heat generated by the high pressure process. And this melting, they can facilitate the binding of the particles, and which increases the tablet cohesion as well as the mechanical strength. And then the second one we have is a liquid to gas, that is vaporization. We will be using them especially in the aerosols. Okay. So in pressurized aerosols, the liquid propellant, it vaporizes rapidly upon the release, and they create a fine mist which delivers the drug to the respiratory tract. Most common example, you can take it as a salbutamol inhaler. And then we have is the solid to gas, that is sublimation of camphor in topical formulations. So here, the sublimation is nothing but the direct transition of solid into the gas, right? So without passing through the liquid phase. So in topical formulations, the sublimation of the volatile compounds like camphor, they provides a cooling effect. So for example, the camphor in balms and liniments that we have, it sublimates upon the application, which releases the vapors, and that is the reason you feel a very cooling and soothing effect on the skin. So what are the benefits for this type of the transition? It enhances the patient comfort by providing a cooling effect, and also it helps in relieving the pain and as well as the inflammation in the topical formulations. And then the next one we have is the techniques for studying the drug properties we have. So the first one is polymorphism, that is to determine what is the type of a polymorph that is present. You can determine it with XRD, which is nothing but X-ray diffraction studies, and DSC, differential scanning colorimetry. So this XRD, it is mainly used to determine the crystalline structure of a compound, and it identifies the polymorphic forms by analyzing the diffraction patterns of X-ray, which is passing through the crystalline substance. And then the applications here, like they differentiate between the stable as well as the metastable polymorphic form of a drug. And then DSC, it measures the heat flow associated with that of the phase transitions. That means what is happening when melting is happening? It is absorbing the heat. What happens when vaporization is happening? It is again absorbing the heat. What is happening when freezing is happening? Okay. It releases the energy. Like that, it is going to measure the heat flow which are associated with the phase transitions. It mainly identifies the polymorphic forms based upon their unique thermal behaviors, right? And then vapor pressure. To measure this vapor pressure, we have TGA, thermogravimetric analysis. We have thermogravimetric analysis. So it measures the weight changes of a substance as a function of its temperature and the type. And it determines the vapor pressure by observing the loss of the volatile compounds. Like, for example, it evaluates the thermal stability as well as the solvent evaporation in the formulation. And pKa determination, that is the dissociation constant, how you would determine it by two titrations, that is potentiometric titration and the UV visible spectroscopy. So the potentiometric titration is, it determines the pKa by measuring the pH changes, like base or acid is added to the drug solution. So this is the accurate pKa determination for weak acids as well as the bases. And UV spectroscopy, we use that, which monitors the changes in the UV absorbance with pH in order to determine the pKa value. And it is mostly suitable for the drugs with UV absorbing functional groups, right? And then we have the regulatory implications. So it has a role in quality control, drug product design, as well as the regulatory submissions like IND and NDA, investigational new drug and new drug application. So what is its role? The states of matter or the physicochemical properties of the drug molecules, what is its role in the quality control? In the quality control, it is going to ensure the consistency as well as the compliance with that of the pharmacopoeial standards. And also the techniques like XRD, DSC, they helps to verify the drug purity as well as the stability during the manufacturing. And coming to its role in the drug product design, it helps to select the appropriate formulation strategies and also to enhance the drug stability as well as the bioavailability. And it is also going to predict the drug's behavior under various environmental conditions. And its role in regulatory submissions is the detailed characterization of the states of matter as well as physicochemical properties is required in most of the regulatory dossiers. Okay, and this data supports the claims about whatever you are claiming, like my drug is stable, my drug is safe, my drug is effective. So all this data of the physicochemical properties of the drug molecules, it forms as a proof for what you are claiming your drug as. Okay. And then coming to the conclusion here, always understanding the states of matter and the physicochemical properties, it is essential to design a stable, effective, and patient-centric drug formulation. And also, it addresses the challenges like poor solubility, low bioavailability, as well as the instability. And it is also useful in designing the stable and effective dosage forms, right? And then both integration of the theoretical and practical knowledge, it is essential for the successful product development. That means it is not only like today you are listening to the class and tomorrow if you're not applying the same in the practical, that it is of no use. So you have to combine the theoretical insights of phase transitions and ionization constant with practical techniques like XRD, DSC, and TGA. It helps in ensuring the successful development of the drug. So the states of matter as well as physicochemical properties of the drug, they it should have a strong foundation for addressing the regulatory, manufacturing, as well as the therapeutic challenges, right? So one should have a brief knowledge about the states of matter and the drug properties, which enables for the innovation as well as the excellence in the pharmaceutical formulation and the product development. This is all about the states of matter and then the physicochemical properties of the drug molecules, right? Hope you all have understood it thoroughly. If you have any doubts regarding today's content, please feel free to post your questions in the comment section below. We are always ready to help you in clarifying your doubts. Thank you so much, everyone, for joining today's session. Thank you.